Welcome back to the deep dive. Our mission here is, well, it's pretty simple. We take a stack of dense, often intimidating sources, and we try to extract the absolute core insights to turn all that complexity into mastery-level knowledge for you.
Today, we're really going to dig into a condition that's been recognized for, nearly 2 millennia, thyroid enlargement, or as it's known, goiter.
Right.
And it's a subject that I think truly illustrates one of the most compelling narratives in biology, this really intricate dynamic interplay between our environment and, our own genetic blueprint.
Absolutely. The superficial understanding often just stops at, oh, it's an iodine issue. But our focus today is to go way, way beyond that. We're plunging into this complex web of what actually causes it, the specific environmental threats, the hard data we're seeing from twin studies, and the molecular mechanism, the pathophysiology that drives the formation and progression of the benign multinodular goiter.
Exactly.
We're aiming for a complete mastery of this specific disease state, and we're relying entirely on the scientific evidence provided.
So to start, we really have to establish a baseline. What scientifically is a goiter today? This definition has evolved a lot alongside our technology.
I can imagine.
Historically, based on these older autopsy studies, a thyroid was classified as enlarged if its weight was more than 35 grams.
Wow. Okay. That's a very blunt postmortem measure. How has modern tech sharpened that definition?
Precisely. Yeah, the shift to high-resolution ultrasound has just revolutionized how we assess the gland in vivo. Today, the accepted definition is based purely on volume.
Okay.
So a thyroid volume greater than 18 milliliters in women and greater than 25 milliliters in men. This shift allows for much, much earlier detection and, better tracking of how well interventions are working.
But I imagine setting those exact cutoffs has to be a challenge when you're comparing different populations around the world.
It's a huge methodological challenge. While those 18 and 25 milliliter figures are pretty standard in a lot of places, variations in these specific volume cutoffs exist all over the globe.
So you have to be careful when comparing studies.
You do. If you're comparing a study from Europe to one from Asia, you have to be really cautious about whether they used exactly the same threshold, because even a slight difference can complicate any direct comparisons of prevalence.
Speaking of prevalence, before these big iodine programs, just how common were thyroid nodules?
Well, before mandated global iodine programs became widespread, those older autopsy studies were reporting the prevalence of thyroid nodules in clinically normal thyroids to be in excess of 50%.
50%. So more than half of us anatomically just had them?
That was the thinking. But that figure is probably an overestimation for today. The current thinking, which is backed by modern ultrasound, suggests the incidence is more realistically between, say, 25 and 50%.
That's still a massive range.
It is, but it tells you that even today, maybe a quarter to a half of clinically normal individuals might have these anatomical variations. It just signals how incredibly prevalent this condition is.
Okay, that's an incredible baseline. Now, before we drill into the causes, let's make sure we're focused. We said we're looking specifically at the benign multinodular form.
Yes. So we classify goiter as either physiological or pathological. Physiological goiter is often transient. We see it in teenage girls, for example. And it usually doesn't have any clinical significance. It just resolves on its own.
And pathological.
Pathological goiter, on the other hand, needs attention. And it includes both benign and malignant forms. For this deep dive, our exclusive focus is on the benign multinodular goiter. This is a condition where the gland not only gets bigger, but also develops multiple non-cancerous lumps. When you see one that's been surgically removed, the gland is characteristically just highly irregular and heterogeneously nodular.
Okay, let's unpack this. When we talk about what drives goiter, we have to start with the single biggest contributor, the absolute foundation of the problem, iodine deficiency. And this isn't just some historical footnote. The scale of this problem today is just staggering.
It really is. The World Health Organization, or WHO, estimated back in 2004 that 2 billion people worldwide were iodine deficient.
2 billion?
2 billion. To put that in perspective, that's almost 30% of the entire global population at the time. And critically, this figure included about 285 million school-aged children, which is the demographic that's most vulnerable to the developmental effects.
And it's not just restricted to the developing world, is it? I think we often assume the Western world has this completely fixed.
And that's a dangerous assumption. If you just look at Europe, around 45% of Europeans are currently thought to be iodine deficient.
Wow, nearly half.
Nearly half the continent is facing some level of functional deficiency, which, it explains why goiter is still endemic or regularly occurring in so many regions, despite centuries of medical knowledge.
45% is a public health crisis just waiting to happen. So, to measure this effectively, what are the hard numbers? What defines enough? And how do you even measure this in a population where everyone's eating different things?
Right. So first, the requirement for non-pregnant adults is about 150 micrograms per day. The major sources are marine life, so fish, seaweed, and also dairy products, which reflects the high iodine content in coastal soil that gets into the food chain.
Okay. And the measurement.
To measure this across a whole population, researchers rely on a key surrogate marker, the urinary iodine concentration, or UIC.
Why urine?
Because about 90% of the iodine you ingest is excreted in your urine. So the measurement of UIC gives you the most accurate, non-invasive reflection of your recent iodine intake. And critically, this measurement is usually corrected for creatinine excretion, which helps to account for variations in how diluted the urine samples might be. It gives you a standardized measure.
That makes sense. So that's the how. Now, what are the actual numbers that define deficiency? Because this has to be the framework for all those global health programs.
This is really the bedrock of any clinical or policy intervention. We define iodine sufficiency as a urinary iodine concentration greater than 100 micrograms per liter.
Okay, 100 is the magic number.
100 is the target. The deficiency states are broken down systematically. So mild deficiency is a UIC between 50 and 99. Moderate deficiency is a UIC below 50. And severe deficiency is the most critical. That's defined as a UIC below 25 micrograms per liter.
That framework is the first step to mastering this. The second, I guess, is understanding this critical, almost perfect inverse relationship between those levels and the size of the thyroid itself.
Indeed. Yeah, the analysis of the epidemiological source data shows this relationship so clearly, and it's the key reason why iodine supplementation just works. To put it simply, as iodine intake goes up, which we see as a rise in median urinary iodine excretion, the median thyroid volume measured by ultrasound falls.
So it's a direct correlation, more iodine, smaller thyroid.
It's a statistically powerful inverse correlation. The points in the graph just show this steep negative decline. It's beautiful data. And what's more, increased UIC also negatively correlates with thyroid nodule prevalence. So fewer nodules develop when iodine status improves.
But the importance goes way beyond just anatomical size. We have to talk about the clinical consequences, especially in school children, which is where the data is most definitive and, well, the effects are most severe.
Absolutely.
So let's break down those severity levels, maybe starting from the worst case scenario and working our way up based on the WHO's categories.
Okay. So at the most critical end is severe iodine deficiency. That's defined by a median urinary iodine excretion below 20 micrograms per liter. In populations at this level, the goiter prevalence among school children is over 30%.
Then the consequences.
They're catastrophic and often irreversible. We see endemic cretinism, which is a severe congenital hypothyroidism that leads to developmental disabilities. We see overt hypothyroidism in the general population, and widespread endemic nodular goiter. This is the classic picture of a population just struggling for basic thyroid function.
That's the devastating historical outcome. Moving up to moderate iodine deficiency, where the UIC is between 20 and 49 micrograms per liter. The picture changes a bit, but it's still dire.
Correct. Goiter prevalence is still alarmingly high, between 20 and almost 30%. The associated issues still include endemic nodular goiter and, critically, a measurable decrease in intellectual quotient, or IQ.
Wow.
But what's really fascinating here is the shift in the hormonal pathology. At this moderate level, you actually start to see hyperthyroidism, an overactive thyroid, appearing more often than hypothyroidism.
Wait, why would that happen? If the population is still deficient, why does overactivity suddenly emerge?
It's a great question. The gland is struggling to produce enough T4, but the continuous compensatory stimulation it's getting from the pituitary gland, which is desperately trying to normalize hormone levels, it starts to drive certain susceptible areas of the thyroid into autonomously functioning states.
So it's like patches of the gland go rogue.
Exactly.
Yeah.
And start producing hormone regardless of what the body actually needs.
That explains the shift in how the disease shows up. You're moving away from just pure insufficiency towards this potential for autonomous overactivity in parts of the gland.
Precisely. And now, even in mild iodine deficiency, where the UIC is between 50 and 99, goiter prevalence is still a major concern, ranging from about 6 to 20%. At this level, hyperthyroidism often definitively outweighs hypothyroidism as the dominant clinical concern, and nodular goiter is still very clinically relevant.
So even what we call mild deficiency is still enough to drive nodules and specific harmful hormonal issues. When does the prevalence finally drop to an acceptable level?
Only when a population reaches iodine sufficiency, so a UIC between 100 and 199 micrograms per liter. That's when the goiter prevalence finally falls to a minimum, less than about 5%. This is really the optimal balance point.
Given these profound consequences, it seems obvious that every country would enforce correction globally.
Well, the primary most effective policy is mandatory salt iodization. It's simple, it's cheap, and it's undeniably effective at raising UIC levels across a population. But fundamentally, it's not universal, and this creates these persistent public health vulnerabilities even in the wealthiest nations.
Let's use the United Kingdom as our case study here because the data is incredibly sobering. It really contradicts that assumption that all developed nations have this handled. We have a 2011 national survey that looked specifically at female schoolchildren aged 14 to 15. What did it find?
The data is a major wake-up call. They found the median urinary iodine concentration in that cohort was 80.1 micrograms per liter.
80.1. So if we remember our definitions, that falls squarely in the category of mild iodine deficiency.
Exactly. And that classification meant that a staggering 67% of that cohort, nearly two-thirds of teenage girls, were iodine deficient, and 17% were classified as moderately or severely deficient. This confirms a persistent, significant public health burden right in the middle of a highly developed Western nation.
That fact alone should trigger major policy action, especially when you consider the intergenerational risk. I mean, the consequences go far beyond just a slightly enlarged neck for one person.
The long-term impact on cognitive function is where the true cost really lies. A detailed study looked at mother-child pairs in the UK. They measured maternal UIC during the first trimester of pregnancy and then correlated that iodine status with the child's cognitive development at age 8.
And what did that study reveal about the long-term cognitive costs of just mild deficiency during that critical developmental window?
The core finding was that even mild iodine deficiency in early gestation was associated with measurable, lasting deficits in specific cognitive areas by age 8. We're not talking about profound, obvious disabilities here, but measurable reductions in function.
Can you break down those specific deficits for us?
Yeah, so the children who are exposed to mild deficiency in utero scored lower in 3 really fundamental cognitive areas: verbal IQ, reading accuracy, and reading comprehension. And if we look specifically at the verbal IQ data in the sources, you can see a clear dose-response relationship. When the maternal iodine to creatinine ratio was below 50 micrograms per gram, indicating deficiency, the average verbal IQ score was significantly lower compared to those whose mothers had a healthy ratio above 1 to 50.
So this emphasizes that correcting a trace element deficiency is one of the single greatest tools we have for raising the collective cognitive function and intellectual potential of a population.
It is. It's a profound long-term risk that spans generations. And it makes iodine deficiency correction just about the most impactful public health measure we can take against goiter and all its consequences.
Okay, so while iodine deficiency is clearly the dominant factor, the macro-environmental stress, we have to move on to other environmental factors and metabolic influences.
Right, and this is a nuanced area. It's critical to stress that the impact of these other goitrogenic factors is almost always examined in the context of the underlying iodine status.
So, iodine deficiency acts as a kind of vulnerability amplifier. When iodine is low, the thyroid is already working overtime, which lowers the threshold for other environmental insults to cause damage.
Precisely. Their effects can be really significant when iodine is depleted, but they might be minimal or even completely absent when a population is iodine sufficient.
Let's start with the chemical threats, these goitrogenic pollutants that interfere directly with how the body uses iodine. What about the chemical that's often linked to cigarette smoke, thiocyanate?
Right, so thiocyanate is a major goitrogen. It interferes with the thyroid gland's ability to trap and concentrate iodide. We encounter it in 2 primary ways that are, critical to population health. First, as you said, it's a key component of cigarette smoke, which immediately flags tobacco as a risk factor.
And the second?
Second, and this is crucial for vulnerable populations, it's released when consuming cassava. Cassava is a major starchy carbohydrate source in parts of Africa, and high consumption is directly associated with an increased incidence of goiter in those regions, especially where iodine is already scarce.
That's a perfect example of diet interacting with geography and existing nutrient status to drive disease. What about contaminants that get into the food chain through water?
Next up is nitrate. Sources include certain vegetables and, critically, drinking water, especially in agricultural areas where you have high contamination from runoff. The research here is really interesting because the findings are a bit mixed, which just highlights the challenges of these environmental toxicology studies.
Can you contrast the data for us?
Sure, in a study looking at school children in Bulgaria, a population that was considered iodine replete, so their iodine levels were generally okay, contamination of the water supply with nitrate led to a really significant finding. The odds ratio for goiter was 3.
So it tripled the risk.
It tripled the risk of goiter formation. But in contrast, a major German study found no definitive link between urinary nitrite excretion and measured thyroid volume in their population.
Which suggests
It suggests that the impact of nitrate may be highly dependent on regional factors, the type of contamination, or maybe interactions with other goitrogens in that specific environment.
it really shows that environmental science is rarely black and white. There are always these other factors at play. What about other plant-based compounds, like the ones in soybeans?
Yes, we have to talk about isoflavones, which are plentiful in soybeans. These compounds are known to chemically inhibit thyroid peroxidase, which is the essential enzyme for iodinating the thyroid hormone precursors.
And what does the human data say?
Again, when you look at large-scale human population studies, the results have been mixed, likely because they're muddled by the complexity of our diets and varying iodine intake.
But the underlying biological mechanism is clearer in a controlled setting.
Absolutely. When you use laboratory models, specifically mouse models that are fed a soybean-rich diet, the research consistently shows an increased thyroid size. This suggests that while the human population data might be masked by other variables, the fundamental biological mechanism for goiter formation is definitely there with high isoflavone exposure.
Okay. Before we move on, what about some of those older solvents and pesticides?
You're talking about organic chlorides. These are solvents and pesticides, many of which are now banned or heavily restricted, so their prevalence in the environment is going down. They're generally considered to have only a mild goitrogenic effect, and importantly, this risk is expected to continue to decline.
Ivory, let's talk about perchlorate. The name sounds potent, rocket fuel, bleach, but the source material makes a crucial distinction about its actual clinical impact at common environmental levels.
This is a really crucial distinction that often causes unnecessary public anxiety. Perchlorate is theoretically a potent inhibitor of iodine uptake. You find it in rocket fuel, certain bleaches, and fertilizers. And while the U.S. Environmental Protection Agency, the EPA, does monitor and regulate perchlorate in water, the clinical reality is pretty reassuring.
And what is that clinical takeaway?
The environmental exposure levels have generally not been shown to affect thyroid function clinically. They're very unlikely to affect thyroid volume in the general population because the body has this remarkable capacity to compensate.
So the dose makes the poison, basically.
Exactly. The critical distinction is that the EPA's cutoff for detectable levels is many, many orders of magnitude lower than the concentration you'd need to cause any measurable clinical thyroid effects. So while it's a chemical inhibitor, it's generally not a primary driver of goiter at the low, typical environmental concentrations we see today.
That puts the chemical risk into proper perspective. Let's move to lifestyle, starting with tobacco, which brings us back to one of the goitrogens we just discussed.
All right. Tobacco smoking is identified as a major consistent risk factor, especially, and this is key, in iodine-deficient populations. The odds ratio for developing goiter in smokers, compared to non-smokers, ranges between 1.5 to 3.
And the mechanism is the thiocyanates.
It's almost certainly tied directly back to the thiocyanates we talked about earlier, which are major constituents of tobacco smoke, and directly inhibit that iodine uptake.
And what about other micronutrients? I know selenium comes up a lot in the literature.
Selenium plays a vital supportive role. While iodine is the raw material, selenium is necessary for the final stages of thyroid hormone synthesis. But more importantly, selenium is crucial because it's required for the function of selenoproteins, which are like the gland's internal cleaning crew.
Why does the thyroid need a cleaning crew?
Because the thyroid peroxidase enzyme, the one doing the essential work of iodinating, generates a significant amount of hydrogen peroxide, H2O2, as a natural byproduct.
Which is a free radical.
Exactly, a reactive oxygen species. Selenoproteins help manage and neutralize it. Therefore, selenium deficiency plays this cumulative role. Alongside iodine deficiency, a lack of selenium just exacerbates the oxidative stress, contributing directly to the cell damage and mutation load that drives nodule formation.
Okay, now we get to two of the most counterintuitive findings in the source material, starting with the effect of the oral contraceptive pill, or OCP.
Yes, Since we know hormones influence goiter, and goiter is much more common in women, your intuition might suggest that OCP use would increase the risk.
Right.
But the opposite appears to be true. OCP use is associated with a lower thyroid volume, on average 8.5 milliliters compared to 9.4 in non-users, and a statistically significant low odds ratio of 0.6 for goiter development.
That's a measurable protective difference. What's the leading theory for this counterintuitive benefit?
The precise mechanism is still unexplained and needs more research, but the leading hypothesis is all about estrogen regulation. The exogenous estrogens from the pill may suppress the body's own endogenous estrogens. And since endogenous estrogens are thought to be mildly goitrogenic themselves, the OCP essentially provides a suppressing mechanism, reducing the overall hormonal drive for enlargement.
That's fascinating. The second surprising finding, and one that is becoming more and more relevant, links goiter directly to metabolic syndrome and insulin resistance.
This connection is a major area of current research, mainly because of the worldwide rise in obesity and type 2 diabetes. Insulin resistance has been identified as a strong risk factor for nodular goiter.
And the data.
The data is pretty stark. In areas already struggling with some iodine deficiency, nodules were found in 53% of patients with insulin resistance, compared to only 19% of patients without it.
That's nearly 3 times the risk. So insulin resistance, a systemic metabolic problem, seems to be accelerating the formation of thyroid nodules. Has this led to any novel treatment ideas?
It has. It immediately pointed researchers toward existing metabolic drugs. The well-known diabetes drug metformin, which improves insulin sensitivity, has shown some positive effects in reducing thyroid volume in these specific patient groups.
By normalizing the local hormonal environment.
That's the theory. But these results aren't yet conclusive across all the studies. So while it's a very promising avenue, we can't yet confirm its place as a standard independent treatment for goiter.
What's fascinating here is that while we've established iodine deficiency as this overwhelmingly dominant environmental driver, the fact that goiter so often clusters within families, plus its higher prevalence in females, it just screams that there's a profound underlying genetic component.
Absolutely. Even in iodine-sufficient populations today, some families continue to show high rates of thyroid volume increase, which provides really compelling evidence of an inherent genetic cause.
So how do researchers definitively separate the genetic risk from the environmental risk when, everyone in a family generally shares the same diet and local environment?
The gold standard for providing direct, hard evidence of a genetic basis is the use of twin studies. These studies compare the concordance for goiter development between monozygotic, or MZ, twins, identical twins who share almost 100% of their genes, and dizygotic, or DZ, twins, fraternal twins, who share about 50% of their genes, but have the same prenatal and childhood environment.
And what does the twin data definitively tell us about the power of genetics here?
The concordance rates are striking. They're the most powerful proof we have. In areas where goiter is endemic, so the environment is severely iodine deficient, MZ twins show a concordance rate of 80%.
80%.
If one has goiter, the other has an 80% chance of having it. DZ twins in that same environmentally stressed area only show about 40 to 50% concordance. The shared genetics clearly exert a dominant influence, even when the environment is hostile.
That's powerful. And what about in areas where that environmental stress is reduced?
In sporadic areas where iodine deficiency isn't endemic, the genetic influence is still clearly dominant, though the overall rates are lower. MZ concordance drops to 42%, but that is still way higher than the mere 13% you see for DZ twins.
So no matter what, the identical twins are much more likely to share the condition.
Exactly. The significantly higher rate in identical twins, regardless of the iodine environment, proves genetics plays a major constant role.
But the relative influence of genetics versus environment seems to shift dramatically based on how severe the environment is. We've got specific data that quantifies this precise balance.
This is a really critical insight into the interplay. We can see the thyroid's adaptive strategy in action. When we look at data adapted from older studies in Scotland in 1967, a time of severe iodine deficiency, the environmental factors totally dominated goiter development. They contributed 61%, with genetics only contributing 39%. The environmental stress was the primary overwhelming force.
So when the environment is severely challenging, the thyroid's function is dictated by that external problem, and the genetic predisposition kind of takes a back seat to sheer necessity.
Exactly. But look what happens when the environment improves. Studies from Denmark between 1999 and 2005, where the population was only mildly deficient, showed a radical inverse shift. In 3 separate analyses during this period, genetic factors contributed a strong majority, ranging from 71 to 82%, while environmental factors only contributed 18 to 29%.
That is the crucial clinical takeaway then. As environmental stress, specifically iodine deficiency, is fixed by public health policy, the inherent pre-existing genetic susceptibility becomes the overwhelmingly significant driver of who develops goiter and who doesn't.
That's it. The genetics are revealed once you quiet the environmental noise. It explains why a policy of universal iodine supplementation will fix goiter for most people, but certain families will still struggle.
Right. And that leads to the question, what are these specific genes? Let's start with the thyroid-specific genes, the core machinery of the gland itself.
So the search here focuses on genes we know are involved in the normal process of making thyroid hormone. And the mechanism is simple compensation. Mutations in these genes generally lead to suppressed or impaired thyroid hormone production. The body senses these low hormone levels, and the pituitary gland increases its signaling, telling the thyroid to enlarge in a futile attempt to compensate.
So it's basically the gland trying to overcompensate for a broken part in the assembly line. What are the key genes that have been identified?
Well, mutations in the thyroglobulin, TG gene, are typically linked to a hypothyroid state. Thyroglobulin is the protein backbone that thyroid hormones are built on, so if that's faulty, you have a problem.
And the essential enzyme?
That would be the thyroid peroxidase, TPO gene. This enzyme is crucial for attaching the iodine atoms to the thyroglobulin. Mutations here are also often linked to hypothyroidism, although sometimes patients can remain euthyroid, meaning they maintain normal hormone levels despite the anatomical defect.
Okay.
Then we also have mutations in the thyroid-stimulating hormone receptor, TSHR gene, the pendrin, PDS gene, and the sodium iodide symporter, NIS gene, which is the cellular pump that brings iodide into the cell. All these mutations disrupt that delicate functional balance, forcing the thyroid to enlarge itself.
So those genes govern the gland's direct function, but the source has also identified genetic candidate areas outside that core machinery. These are the non-thyroid specific loci.
These are arguably the most interesting genetic findings because they show that general growth control plays a role. These loci were identified through genetic linkage analysis, which basically tracks the inheritance of specific chromosomal regions in families with the disease.
And where did this analysis point?
It pointed to several candidate regions, which they've dubbed MNG loci, for multinodular goiter. We have MNG1 on chromosome 14q31, MNG2 on XB22, and MNG3, which is localized generally to chromosome 3. Other specific loci were also found on 2q, 3p, 7q, and 8p.
So what's the significance of finding these regions that aren't directly related to making hormone?
The significance is that these loci represent potential genetic switches that control general cell growth or responsiveness to growth factors, separate from the thyroid-specific hormone-making job.
That's a lot of different potential locations across the genome, which I guess leads to the final genetic conclusion.
Exactly. While these discrete loci exist and they're important targets for future research, the consensus conclusion remains that the vast majority of benign goiter cases are likely polygenic, meaning multiple different genes work together to increase susceptibility, and they are all heavily modulated by the environment, with iodine status being the primary gatekeeper.
And all of these factors ultimately feed into the pathological model of how the gland actually transforms, which is our next crucial topic.
If we connect all these environmental and genetic factors to the bigger picture, the development of a benign multinodular goiter is best understood through a specific two-phase or bimodal model. This model perfectly delineates the transformation of a normal gland into a heterogeneous, lumpy mass.
Okay, so let's walk through how this actually happens inside the body. Let's start with phase 1, the initial trigger and the body's attempt to compensate.
Okay, so phase 1 is the goitrogenic stimulus and global proliferation. The trigger, as we know, is often iodine deficiency, which means the gland can't make enough T4. The body's immediate adaptive response is global hyperplasia, an increase in the number of thyroid cells and/or hypertrophy, which is an increase in cell size.
So the gland is just trying to get bigger to do its job better with limited supplies?
That's it. The entire gland swells in an effort to maximize its functional capacity, essentially trying to squeeze more hormone out of limited resources.
But you mentioned earlier that this adaptation comes with a huge internal cost.
It does. This increased rate of hormone synthesis, and the growth that goes with it, requires the thyroid peroxidase enzyme to work overtime. And as we discussed, this generates significant amounts of hydrogen peroxide and other free radicals.
It's like running an engine in the red for too long. Things start to break.
That's a great way to put it. The constant oxidative stress results in crucial cellular damage, what we call wear and tear. And that wear and tear translates directly into a higher risk of genetic mistakes. The oxidative stress results in a crucial increase in spontaneous mutation rates throughout the entire gland, creating what we call a very high mutagenic load.
Okay, so that's the setup for the actual formation of the lumps. Let's walk through phase 2, nodule formation and clonal expansion.
Right, so this high mutagenic load massively increases the chance of randomly generating somatic mutations. Mutations that happen in a single cell after you're born that specifically give that cell a growth advantage. These are the lucky cells that get a genetic superpower telling them to keep growing no matter what.
And those specific mutated cells are also being bathed in a stimulating environment.
Correct. The entire proliferating area is saturated with local growth factors. Things like insulin-like growth factor, IGF, transforming growth factor, TGF, and epidermal growth factor, EGF. They all stimulate the division of the surrounding cells, including the ones that are already mutated, which rapidly accelerates their development.
So what happens if you fix the iodine problem at this stage? Is it too late?
This is the absolute key step in understanding why goiter persists even after you fix the problem. When that initial goitrogenic stimulus is removed, say iodine is replenished, the proliferation of the normal thyrocytes stops. They go back to a healthy, quiet state. But the activated cell clones that acquired those growth advantage mutations, they continue to proliferate autonomously. They are now independent of the body's normal regulatory feedback loops, and they just keep dividing, forming these discrete, palpable nodules.
And that's why you get a lumpy goiter, not just one big smooth one, because it's different mistakes happening in different cells.
Absolutely. The result is nodule heterogeneity. The goiter is a polyclonal entity, meaning it originated from multiple separate cell lines that acquired different independent mistakes. And the specific nature of the mutation dictates how the nodule behaves.
We need to unpack that distinction between the different kinds of nodules, the hot and cold lumps, and how they relate to the underlying cellular machinery.
This is where we look at the specific pathways the mutations activate. So if a mutation activates the cyclic AMP or CAMP cascade, that cascade typically leads to both growth and function. This results in an autonomously functioning nodule, which is often called a hot nodule because it produces thyroid hormone independently of any stimulation.
And what about the non-functioning or cold nodules?
If the somatic mutation instead activates the mitogen-activated protein kinase, or MAPK, pathway, this specific pathway primarily leads to proliferation only. So these cells are aggressively multiplying, but they aren't contributing to hormone output. The difference between these two signaling pathways accounts for the highly varied clinical appearance and function you see in all multinodular goiters.
That bimodal model gives us a roadmap from environmental insult all the way through to the fully formed heterogeneous disease. Now let's shift focus to the complex hormonal control that governs all of this, beyond just iodine availability.
Right, the control system is incredibly intricate. You have endo, auto, and paracrine pathways. But the central undisputed figure in regulating thyroid growth is the thyroid stimulating hormone, or TSH. TSH is the principal stimulator of thyroid cell growth.
And how does TSH actually signal to the thyroid cell?
Its receptor, the TSH receptor, is a really important protein on the basal membrane of the thyroid cells. It belongs to the G protein-coupled receptor, or GPCR family. Think of GPCRs as the most common fundamental communication switchboard in our cells.
Okay.
When TSH binds to this switchboard, it activates cellular processes, primarily the adenyl cyclase CAMP system, which ultimately promotes cell growth and function.
So TSH turns on the growth signal. But we have this fascinating counterintuitive data point, this TSH paradox, about its action at very high concentrations.
Yes, the TSH paradox is conceptually similar to the Wolff-Chaikoff effect, where high levels of iodine can transiently inhibit thyroid function. In a similar way, high serum TSH concentrations have been shown to have an inhibitory effect on thyroid cell proliferation in vitro in laboratory cultures.
Wait, so too much TSH can shut things down? That seems to fundamentally contradict its primary role.
It does, and it suggests there's a complex internal regulatory switch, probably an ancient evolutionary mechanism, that prevents runaway proliferation even when there's an overwhelming stimulating signal. The source data from cultured thyroid cells illustrates this perfectly. At normal to moderately high TSH concentrations, iodide uptake is stable. But when you raise the TSH concentration dramatically, to 10 units, for example, the relative iodide uptake by the cells just plummets. It's a high-dose inhibition, a regulatory failsafe.
Beyond TSH, we established that there's this soup of local factors promoting the clonal expansion of those rogue cells. We need to detail this complex local growth factor signaling network.
This network is key. It controls not just cell growth, but also angiogenesis, the formation of new blood vessels, which the nodules need to get any bigger. And these factors act through several distinct signaling cascades.
Let's start with the one most closely tied to TSH and function.
That would be the adenyl cyclase CAMP protein kinase A PKA pathway. As we mentioned, this is the primary recipient of TSH signaling, although TSH also has that strange high-dose inhibitory action. This pathway is also stimulated by epinephrine.
Then we have the largest group, which includes many of the factors we hear about in general cancer research, right?
Exactly. That group operates through receptor tyrosine kinases. This is a major fundamental pathway for growth factors throughout the body. The list is extensive. It includes epidermal growth factor, EGF, transforming growth factor alpha, TGF-alpha, various fibroblast growth factors, FGFs, insulin-like growth factor I, IGF-I, hepatocyte growth factor, HGF, and platelet-derived growth factor, PDGF. All of these act locally to stimulate proliferation.
And finally, there's a pathway that seems to balance stimulatory signals with inhibitory control.
That's the phosphatidylenoacetyl cascade. This pathway is stimulated by TSH and by chemical agents called esters. But crucially, it's inhibited by two specific factors: transforming growth factor beta, TGF-beta, which is often an internal growth-suppressing signal, and, strikingly, by iodides.
So iodide itself is an inhibitor here.
The fact that iodide, the essential trace element, is listed as an inhibitor in this critical growth cascade just underscores its multifaceted role. It's not just a raw material for making hormone, it's a direct regulator of cellular growth. It tells the cell when to slow down.
Understanding this intricate interplay is vital for designing new treatments. Currently, what is the default treatment for a large multinodular goiter?
The current mainstay for managing both benign and malignant thyroid disease, especially when the goiter is large enough to cause compressive symptoms or cosmetic issues, is surgery, a thyroidectomy.
But the source material emphasizes a major clinical gap here, particularly on a global health scale.
Absolutely. We have to acknowledge that globally, for many patients who desperately need treatment, surgery is just not an option. This could be because they're not suitable for general anesthetic, or far more often in developing countries, there's a critical lack of access to the right surgical facilities and expertise.
Which means we desperately need ways to shrink these things without putting people under the knife.
Exactly. This emphasizes the dire global need for non-surgical treatment modalities.
So where is the cutting-edge future research focusing to fill this massive clinical gap?
Current lab studies are focused heavily on gene targeting, specifically on inhibiting the growth factor receptors that drive proliferation and angiogenesis in those autonomous nodules. They're trying to specifically block the signals that these rogue cells rely on.
Which ones specifically?
The source material specifically mentions targeting vascular endothelial growth factor receptor, VEGFR, which is crucial for forming the blood vessels that feed the growth, and fibroblast growth factor receptor, FGFR, a key driver of cell division. If we can shut down these specific growth signals, we may be able to shrink the goiter without ever needing a scalpel, which would revolutionize global management.
So to summarize this entire deep dive, the formation of goiter goitrogenesis is fundamentally this complex process that results from a powerful interaction between your genetic predisposition and environmental factors.
That's right. And the extent of this interplay is critically dependent on the underlying iodine status. When iodine is severely depleted, the environment just dominates. But once you fix that environmental stress, the underlying genetic susceptibility really takes the lead. And crucially, a detailed knowledge of the involved growth factors and signaling pathways, like the KMP and MEPK cascades, is absolutely vital for the clinician to understand the likely progression of the disease and select the most appropriate treatment.
And despite all the intricate genetics and molecular pathways we've analyzed today, the primary global policy priority remains crystal clear: correcting iodine deficiency. This is still the most impactful, rapid way to reduce goiter prevalence globally, and it remains a key focus of the World Health Organization. But we have to reiterate that the risk of mild iodine deficiency in the developed Western world can't be ignored, as shown by that UK data with its profound cognitive risks in children. Continued efforts to ensure populations get their required intake must be strongly supported.
And looking ahead, the scientific community has some very specific research questions that are going to define the next decade of goiter management and prevention.
First, there's the urgent need for a better understanding of that specific link between metabolic syndrome and goiter formation, a link that just gets more critical with the worldwide rise in obesity and insulin resistance. Second, we need a detailed characterization of the precise role played by those non-thyroid specific genes we touched on, the MNG loci in goiter progression.
And finally, the ultimate goal, which offers hope for patients globally who lack surgical access. The potential use of novel approaches like gene-targeting therapies or recombinant adenovirus treatments for the non-surgical management of large multinodular goiter.
Which leads us to a final provocative thought for you to consider. We know that strong genetic clustering is observed even in iodine-sufficient populations, and we've successfully identified specific non-thyroid-specific goiter loci like MNG1 and MNG2. Given this insight, are we approaching a future where routine, inexpensive genetic screening for these specific MNG loci could preemptively guide lifestyle interventions or medical prophylaxis for those individuals who are deemed genetically highly susceptible? It's a compelling question as we move from a reactive treatment model to a true preemptive prevention strategy.